The seminar. It's my pleasure to introduce to you Professor Koichi Asakura. Asakura Sensei is from Keio University in Tokyo, and actually spent 2 years in this laboratory as a postdoc student. It was a very pleasant stay indeed. And what is studied in this laboratory is, in very general, dissipative structure system that is self-organizing system. And so Koichi will present you all this dissipative structure system, and most especially on they relate to the chiral symmetry breaking problem. So please.
Thank you for your kind introduction, Dr. Frason. I'm from Keio University in Japan, and title of my today's talk is Dissipative Structure and Chiral Symmetry Breaking Transition. I'm staying here for 2 weeks, and during my stay, I will give 3 talks. So today's talk is the most general talk during among 3 talks. So before going into the scientific matter in my talk, let me introduce you my school, Keio. And if you like to travel in Japan, this guy's face is very important, because this guy's face is printed on the highest amount of bill, 10,010. So if you lost this guy's picture from your pocket, you lost a lot. And our school was founded by this person in 1858, and that's why this year is 150 years anniversary of Keio. And these are the histories, and I pick up some important things. And if you travel in Japan, you cannot find any places named Keio, because Keio is the name of the era. You know, we Japan have orange era. Now it's in Heisei era. Heisei means maybe achievement of peace or something like that. And Keio means maybe happy response. Am I right? Dr. Saito, happy response or something like that. Mm-hmm. So after 10 years has passed, Keio has established, and Fukuzawa moves from school to new location, and named that school names Keio. Keio. And what is important is just after 5 years from the establishment of the school, and it is actually the very end of the samurai government era. So learning Western matter, like Western language and Western science and Western social systems, are pretty new for Japanese. But even though it started with Dutch school, Fukuzawa decided to transfer from Dutch school to English school, because Fukuzawa thought that Dutch should be replaced by the English. So that's why 10 years after, Fukuzawa decided to change Dutch school to English school. So design the future is our concept. And we belong to Faculty of Science and Technology. And sorry for small slide characters. And actually, our faculty was established by the individual donation. One of our alumni, Fujiwara, donates to make Fujiwara Institute of Technology, then give it to Keio University. So now we have about 300 faculty members, 4,000 undergraduate students, and 2,000 graduate students in our Faculty of Science and Technology. And not only I, but also Professor Saito came from Japan, also teach at Keio University. Dr. Frason will stay as a postdoc in our university. And let me go into the scientific matter. So at first, starting with a very general matter. Biology reasons are ubiquitous in nature. I'm sorry that some of the topics I'm speaking is very similar to my previous talk, so please be patient if you hear same story like twice. So, spatially periodic patterns are observed in the surface of an animal body. You see, this is a body surface of tiger, and black and white stripes are formed in the body surface of tiger, and it indicates that pigment concentrations are patterns. In black part, pigment concentration is high. In the white part, pigment concentration is low. So this kind of concentration patterns are spontaneously generated in the body surface of animals. And not only spatially periodic patterns, but also temporary periodic patterns are observed in cell cycle, circadian rhythms, and pulsations. And these kind of patterns are very general for living system. But next question is, are these phenomena seen only in life? Because spatially periodic pattern formation and temporary periodic pattern formation looks like a violation second law of thermodynamics. And so these phenomena are seen only in life. Actually, it is. The answer is no. So once you got Pierre Procusion is a person who developed the concept of dissipative structure. And after he developed this concept of dissipative structure, those periodic temporal patterns and periodic stripe pattern, spatial patterns, are not violation of the second law. So he got a Nobel Prize in chemistry in 1977. So let me explain you about the basic concept, what dissipative structure is. So please consider what you are doing right now. You're breathing, so you take oxygen, and maybe you take every 3 meals every day. So you are the system that are open to the other outer environment. You take energy or high chemical potential matter from outside your body, then throw low chemical potential matter from your out from your body. But during this process, metabolism is occurred. So metabolism is a chemical reaction, diffusion, or heat conductions. And these are the irreversible dissipative process. Chemical reactions surely transfer from higher chemical potential matter to lower chemical potential matter. And diffusion surely occurs from higher chemical potential region to lower chemical potential region. And heat conduction occurs from hotter part to colder part. So this is irreversible dissipative process. And these processes surely produce the entropy. And if the system is isolated from the outer environment, those irreversible dissipative processes corrects the fluctuation generated in the reactions, chemical systems. But if the system is open and a system can exchange energy and matter with the outer environment, fluctuation, those irreversible processes make fluctuation grow to a live self-organized state. Self-organized states are oscillations, pattern formation, and chiral symmetry-based transition. So these are the new self-organized state generated in open, far from equilibrium system by the influence of those irreversible dissipative processes. That's why these self-organized states are called dissipative structure. And to maintain this self-organized state, or to generate this self-organized state, those irreversible dissipative processes are necessary. So the entropy production rate is always negative to maintain, generate those cell organic state. So once you understand the concept of dissipative structure, you can make chemical oscillation by general chemical reaction. And you can see the oscillation in the beaker, red and blue oscillations. In this beaker, iron is dissolved in the solution, and when it is red, concentration of Fe2 plus is higher, and when it is blue, concentration of Fe3 plus is high. So the concentration of Fe2 and Fe3 are oscillated as time is passing. And first chemical oscillation was found by Gray in 1921. And Berezov found this Berezov-Zabuzinski reaction in 1958. But most of the researchers among Berezov, he was born and worked in former Soviet Union, ignore his research. So his publication, his article was not published before he died. And these are the chemical experimental reaction. And apart from this chemical reaction, there are theoretical model called Brasseretter. And these two figures are the basic fundamental mechanism of Berezov-Zabuzinski reaction and Brasseretter. So Berezov-Zabuzinski reaction, this bromate is an oxidizing agent, and this malonic acid and brommalonic acid is an organic compound. So during this, by this, very complicated reaction network, oxidizing agent bromate oxidize these organic compounds to make some lower chemical potential product. Mostly, this is mostly carbon dioxide. And BRO3 reducted to HBRO. So it is very similar to the living system. And this is a model called Brasseretter. So in Brasseretter, of course, the system is open to outer environment. So higher chemical potential model A and B flow into the system, and then A and B react to produce intermediate X and Y. And X and Y finally goes to D and E. Those are the final product having having the lowest chemical potential among these 6 species. Then flow away from the chemical reaction system. And in order to analyze the behavior of this system, these 2 differential equations can be formulated. And first one is a rate equation for the concentration of intermediate X, and second one is a rate equation for the intermediate Y. So first process generate X. So first term of this rate equation is K1A. Second is the consumption of X to generate Y. So minus K2BX and K2BX. And third reaction is autocatalytic reaction. In the presence of 2X, Y converts to X. So in this case, K3X square Y and minus K3X square Y. And the final one is the consumption of X, so minus K4X. And so in order to analyze the behavior of these kind of 2 variable systems, linear stability analysis is a good tool. So, for example, dx, both rate equation, rate equation, dx over dt and dy over dt are function of a concentration of x and y. So, in order to see the behavior of this equation, first, you may obtain the steady-state solution when you put this and this equation equal to 0 and solving the regular equation to obtain a steady-state solution. SS means steady-state. Steady-state solution for XSS and YSS. And so if x and y, values of x and y's are XSS and YSS, dx over dt and dy over dt equals 0. So concentration x and y, it doesn't change. But there are two kinds of steady state in the world. For example, this is a stick, and if I put it like this stick is stopped at here. But if you carefully put these sticks on my hand, but if you carefully put this stick on my hand, it stands. But if it's fluctuates a little, this state is close to the other state. So there are two kinds of steady state. One is stable to fluctuation. The other is unstable to fluctuation. So linear stability analysis is the tool to check the stability of this steady state solution to the fluctuation. So perturbation by fluctuation. So if you put x and y as a sum of these steady state solution plus perturbation by fluctuation, then substitute it into this equation. And by Taylor expansion, and then the Taylor expansion was done, and you remain. This is the only first order term of Taylor series. Of course, if you expand Taylor series, there are much more second order, third order, fourth order, higher orders term for the fluctuation. But we only remain this first order term for the Taylor series. The reason why we remain only first order term is delta x and delta y's fluctuation. So levels of fluctuation is usually square root of n if the average is n. So higher order terms are much smaller when it compares with the first order term. So if you know the behavior of first order term, you may guess that the steady state is stable or not. So we put fluctuation as a function time like this. So lambda determines the fate of fluctuation. If sign of lambda is positive, fluctuation grows. But if lambda is negative value, fluctuation decays and go back to the steady state. And so we make Jacobian matrix. It is a matrix for these equations. And you see, eigenvalues of this Jacobian matrix is lambda. So if you check this eigenvalue of this Jacobian matrix, you can understand that the system, that the steady state is unstable or stable. So the characteristic equations are here, and for the so that's why trace of j is, if trace of j is negative and determinant j is positive, the steady state is stable. But in other conditions, steady states are unstable. But this linear stability analysis only available for analyzing the stability of steady state. You cannot know the behavior after the growth of fluctuation because we ignore higher order terms of the fluctuation. But now we have a computer. So we didn't, even though we cannot obtain analytical solution of these non-linear equations, we obtain numerical solutions by simulations. So let me show you some results of the simulation. And this is a simulation for the bracelet, and these are the two sets of kinetic equation for the bracelet. And because for the simplicity, we put those kinetic parameter equal to 1. And initial, and A and B are the concentration of starting compound. And at first, we put these values A equal 1.0 and B equal 1.0. And actually, when those values are like this, the steady state is stable to fluctuation. So please watch this. Watch what happened by simulation. And at first, you obtain. This is a program for obtaining steady state solution. And if those kinetic parameters are 1, and concentration of A and B are 1, the steady state were x equal 1 and y equal 1. And this is a program for obtaining the numerical solution of kinetic equation. And at first, x0 and y0 equal 0. It means initial value of x and y is put this steady state value. And when we make a simulation, of course, both x and y stay at the constant. And when you fluctuate a little, for example, x equal 1.5, so Japanese can 1.5, and in the simulation, even though if we add fluctuation, it goes back to steady state. But when this value change from 1 to 3, and A is 1, and B increase from 1 to 3. So we obtain steady state solution x equal 1 and y equal 3. And here we again put 3. Then start with 1.0 and 3.0. So if x and y values are on the steady state value, it doesn't change. But if we add some fluctuations on here, for example, 1 or something like that, small perturbation is added. If we add small perturbation here, what we obtain is this kind of, after the some induction period, we get oscillating phenomena. And even though if you decrease the value of fluctuation like this, sorry, too small. induction period is getting to be longer, but again, the system is going to the fluctuation, oscillation state. So let me go back to the PowerPoint. And for spatial pattern formation, basic model was developed by Alan Turing. Alan Turing is not a chemist, but he's very famous among the computer scientists. And after 40 years has passed by the proposition of the model, experimental realization was Turing patterns are experimentally realized by French group. Please watch this video. Blue color network. You can see blue color network on this whole chemical reaction system. In blue part, iodide concentration is higher. In this reaction system, starch is dissolved, and when you mix starch and iodide, it has blue color. So those blue part has higher concentration iodide, and white part having lower concentration iodide. And these patterns are spontaneously generated in the solution. And this is a basic Turing model. And a higher chemical potential compound A is flown into the system and converted to the intermediate X and Y. Then finally transferred to the final product P, and this P is thrown out from the system. And this is the initial Turing model. And in this case, kinetic equations are not ordinary differential equation, but partial differential equation, because concentration is not only a function of time, but also spatial dimension. So these two set of equations are required for considering the pattern formation. And these two equations are divided into two kinds of terms. These terms are reaction terms, and these final terms are diffusion terms. So the concentration is changed by reaction and diffusion. So these equations are called reaction diffusion equations. And Turing pattern was established, succeeded in realized in 1990 in French group. They used this gel strip, and in the gel strip, starch is mixed in gel strips, and two reactant solution is flow above and bottom of this gel. Then the reactant diffuse into the gel, and these Turing pattern formations are succeeded. And so in the case of pattern formation, rate equation is not a differential equation, but ordinary differential equation, but partially differential equation. And these are the reaction term, and these are the diffusion term. So, and fluctuation also is not only a function of time, but a function of spatial dimension. So, Jacobian matrix of this two sets of kinetic equation is like this. And trace J is here, and determinant J is here. And in order to generate spatial patterns, this should be negative. Because if this is positive, this system is unstable to temporal fluctuation. So it doesn't go to the steady spatial pattern formation. So in order to generate steady pattern formation, this should be negative. And please watch this next determinant. Determinant is like this. And if it is negative, this system is unstable to spatial fluctuation, concentration fluctuation. But first term is of course square of square q square, so it is positive. And if this is negative, it again unstable to temporal fluctuation. That's why it is again positive. So in order to make this as a negative, this should be positive. But A11 plus A22 is negative. So if A11 is larger than 0, A22 should be, A11 is positive, A22 should be negative. So it means that X is an activator and Y is inhibitor. You remember, A11 is a partial derivative of later generation X by concentration X. So A1 positive means X as X activator of the system. And A22 negative, A22 is a partial derivative of later generation Y by concentration Y. So if it is negative, Y acts as inhibitor. So, and in order to satisfy these two conditions, not only A11 positive and A22 negative, but diffusion coefficient of dy should be larger than dx. So inhibitor should diffuse much quicker than activator. And please remember this. Please watch this figure again. In this case, for CIMA reaction, iodide ion is an activator, acts as activator. And actually, iodide ion form complex with starch. So complex formation of iodide with starch actually decrease the diffusion coefficient in the chemical reaction system. As a result, this kind of spatial patterns are generated spontaneously from totally homogeneous conditions. So once you learn the concept of dissipative structure, you can make this kind of animals, oscillating mouse and traveling wave mouse. You see this color change of the mouse, surface of mouse. At first, it has black color, but 2 weeks later, it is white. And again, 2 weeks later, it's getting black. And please watch the middle. At first, tail and head is black, and center is white. But 2 weeks later, head and tail are white, and center is black. Then again, reversed after 2 weeks. And this is traveling wave mouse. Concentration pigment wave starting from the from the shoulder to the other side.
That's a real mouse.
Real mouse, yes.
They didn't use a pen.
What?
It is not a cartoon.
They did not use a pen.
And it's not my work, but you went to Liken, researchers in Liken make that mouse. And this is how traveling waves are generated in the mouse. From the shoulder it starts and travels through the whole body surface. And I showed you that Berezov-Zabzinski reaction shows some oscillations. But Berezov-Zabzinski reaction not only show oscillatory behavior, but this kind of chemical waves. So if you write pictures on mouse on the top of the picture, chemical waves like Berezov-Zabzinski reaction, the pictures are pretty the same. It's the same. And please, maybe it is hard to.
That's right.
It's not so good resolution. So maybe it is hard to see. In this petri dish, many kind of concentric chemical waves are generated from the some part. You can see. If you don't see it, please come to my computer after the end of this my talk. After the end of my talk. Okay. You see those concentric patterns.
Yes.
Yeah, but now you're
Sorry. Okay, please turn on that, please. And then one more question. Of course, this is not an open system. No energy input or no high chemical potential compound input here. So why this kind of oscillatory behaviors can be possible? So.
There was no special thing in this rotating thing, right?
When it is turned.
Just time.
No special.
Yeah.
The thing what has happened is not like this. Free energy oscillated, and the concentration some compound oscillated between up and down of the equilibrium. What has happened is here. The beaker has excess amount of starting compound, and a starting compound transfer to final compound product to decrease the free energy like this. During this process, intermediate concentrations oscillated like this. So after the completion of the Birolov-Zabzinski reaction without any flow into the chemical system, you get yellow soda, like a Mountain Dew, because it generates lots of carbon dioxide, and the color is yellow, so it is more like a Mountain Dew. And let me go into the chiral symmetry. Sorry for some Japanese, but maybe everybody in this room knows that like your hand, a symmetric molecule has its own enantiomer. Not only natural molecule, but also artificial molecules. It has mirror image. And actually, for considering the origins of life, origins of homochirality is very important, I think. At least I think, because this is alpha helix of protein, and this is double helix of DNA, and these high order structures can be formed because the element of these polymers are homochiral. If amino acid is racemic or deoxyribose is racemic, those kind of high order structure cannot be generated. So origins of homochirality is a very important process for origins of life. And maybe Frank's model is very known model how chiral asymmetry can be generated in the chemical system. And this is the basic Frank's model. And when you see these two sets of kinetic equation, you may imagine this kind of chemical open system. And higher chemical potential A is flowing to the system. Then A is transferred to L and D. And also L is generated by L and D generated by this photocatalytic reaction. And finally, L and D forms lowest chemical potential model to go outside of this system. And if you analyze this Frank's model, at first you obtain this kind of steady state solution. And this is racemic, of course, because SS means steady state. LSS equal DSS means racemic state. But when you analyze its stability by checking the eigenvalues of this Jacobian matrix, one is always positive and one is negative. So this is saddle point. So like a saddle, if you get on a horse, you get on a saddle, like this. So for this direction, it is stable, but for this direction, it is unstable. So it is called saddle point. So one problem for this Planck's model is the system is always unstable to fluctuation. It always goes to chiral symmetry state. So please consider your life environment. You are living on Earth, and Earth is exposed by the energy from the sun. And if you move Earth from away from Earth, what will happen? Now we have current in the ocean. We have so many climate change in the atmosphere. But if energy from the sun reduced below the subcritical level, those convection may be stopped. So if energy coming from the outside from the sun exceed some critical value, these kind of spontaneous generation convection patterns, like patterns in atmosphere and current patterns, are generated in our Earth. So in our field, those are called constraint, and these are called response. That means, please consider open system. Open system, keep the open system away from equilibrium, some chemical energy or energies are required. So, response is, of course, response of the chemical system. So, constraint is the power to move the system away from equilibrium. Then this kind of response, self-organized state, is possible. So next model is developed by Condopuri and Nelson about 20 years ago. This is a modification of Planck's model, and starting compound S and T. Of course, it has higher chemical potential flow into the system, and not only those forward reactions, but also those reverse reactions are considered. Then he analyzed this model to analyze the model, and found out that for example, lambda is a product of concentration of S and T, so it is constrained, and alpha is XL minus XT for XL. Sorry, this is a mistake. This is plus XT. Plus XT. Yes, so it is almost. It is eminently excess. So if this lambda value is below this lambda c, always only one racemic solution is possible. Other solutions is complex conjugate. I forget English. Complex conjugate. So only racemic solutions are real number. But if lambda exceeds some critical value, not only racemic state, but also those chiral asymmetry states are possible. And when you check the stability of racemic state and chiral state, these racemic states are unstable to fluctuation, and only homochiral states are possible. So please watch the simulation again for this matter. So, this is the basic Condopuri and Nelson's model, and S equal 5.0 and T equal 5.0. That means constraint is below the critical value. So when you calculate this steady state, you only get racemic positive real number, XL, XC, yeah, same number, and others negative or complex conjugate. So this is only a steady state. And if you substitute these values into initial values of xl and xt, like here, and check what's going on by simulation, of course, it is stable. Both L and xl and xt are at the steady state level. And when you add some perturbation like 8.2 or something like that, then calculate again, go back to racemic state. But when s equals 6 and t equals 6, this is above the critical value. And if you check the steady state value at first, you see here, xl and xt are different, and xl and xt are different. These homochiral states are possible. Of course, racemic states are still remaining. And check what's going on in the system. At first, xl0 and xt0 is racemic solutions are put on initial value for xl and xt. Then check the behavior. Of course, there's no fluctuation xl and xt at constant. But if you fluctuate a little, for example, maybe this should be 0 or something like that. If you add some values, yes, if you add some, this from this point 1, 2, 3, 4, 5, 1, 2, 3, 4, 9 from 8. This little change, fluctuation, make this bifurcation. So in this, if this value is above the critical value, the racemic system is unstable to fluctuation to go to chiral state. And the point the system alive is actually this point, xl 12.8 and xt 4.8. And if you add these values here and put little fluctuation, it go back to those homochiral state. You don't have much time. This is, okay, let me go back to the PowerPoint. This is the basic contemporary analysis model. And we did some crystallization experiment on this binaftil. And binaftil racemate very, very quickly when it is in molten state. And our crystal and S crystals are separately crystallized when the temperature is about 145 degrees. So when crystallization was done above 145 degrees, and if we start, this kind of bifurcation phenomena was observed because of the secondary nucleation acting as an autocatalysis. But what has happened if you stay liquid until those crystals getting bigger and bigger? And this is a picture taken from the top of the vessel. This kind of vessel, molten states are here, and stay in the supercooled condition a long time. You can see the generation and growth of this crystal. And if you pick up each crystals and check the optical activity, the results are like this. X-axis is a degree of temperature, and y-axis shows enantiomer excess. Both in every cases, plus or minus, enantiomer excess is centered at around 60, and it isn't influenced by the size of the crystal. So we consider this kind of phenomenon. In the supercooled molten states, at the interface of crystal phase, higher chemical potential racemic melt is flowing to the system, then transferred to the chiral crystal, then throw away from the this open system. So we formulated this kind of model. Liquid is here, and liquid is transferred to the chiral clusters on the top of this interface. And during this process, racemic molecule goes to chiral clusters, and chiral cluster go to the chiral interface phase, then go into the bulk phase. So we have this kind of processes, and these kind of four set of kinetic equations are obtained. So G means gross layer, so clusters having S form S configuration, R configuration, and I mean interface layer having S and R configuration. So, but it is very difficult to analyze, so we simplify to ignore these two slope process equal to 0. Then we can obtain this kind of four set of equations. And these four set of equations give symmetric steady state and asymmetric steady state. Of course, this is racemic. And these twos are absolutely homochiral. R is 0 or S is 0. And we see again when autocatalytic process increase to above some critical value, symmetric steady state getting to be unstable, and asymmetric steady state getting to be stable. But actually, this all-one-known phenomena doesn't observed in the real chemical reactions, chemical crystallization system. So we go back to the complex kinetic model. We don't ignore those slope process. And these four equations, we even obtaining steady state solution is very, very hard for this four set of equations. But thanks to the computer, we obtained numerical solutions like this. When B equal slope process, when it is 0, suddenly racemic to totally homochiral state is generated. But when we add small values on those slope processes, those kind of bifurcations are generated. It is more close to the real experimental system. And if you consider spatial inhomogeneity, this kind of relatively wide range of fluctuation can be possible. And this work was mostly done by Dr. Poisson when he was in Japan. And now we have to look at a chemical reaction. And as I told you, Contemporary Nelson's model require some autocatalytic processes in cross inhibition. And one candidate for realization of chiral symmetry breaking in open system is Sawai reaction. In Sawai reaction, when you add final product having some chiral enantiomer excess, the product obtained by reaction has more higher enantiomer excess as shown in this figure. And when you didn't add any chiral compound into the reaction system, enantiomer excess is spontaneously generated as shown in this table. And after those two papers has been published, some groups analyzed why the Sawai reaction can enhance the optical activity. And there are several models was already being published. But very recent paper published by Greenleaf shows more and more complicated behavior. You see, when this SOI reaction was performed in the absence of any color product, resulting in enantiomer that is fluctuated from run to run very much. And if you increase the catalyst, catalyst concentration, fluctuation is getting to be smaller, and finally goes to 100% enantiomer. And those big fluctuations are not only for these enantiomer, but also chemical reaction rate. When reactions was performed by the same reactions conditions, sometimes this reaction suddenly completed within just 20 minutes. But in this case, after maybe 3 or 4 hours has passed, only 6% of the reaction has been completed. So the reactions behavior fluctuated from run to run very much. And maybe next week, in my talk, I'm talking about this phenomenon much more detail. And because 1 hour has been rushing past, so thank you for your attention.
This business with activating inhibitor, do I remember correctly, the activator is the one which actually has a positive eigenvalue, or if positive term on the diagonal?
No.
And the inhibitor, or what is this? When do you call something an activator and an inhibitor?
This is a Jacobi matrix, and A11 is a partial derivative of F is a rate of generation X. So if this rate was derived by X and positive, it means if the concentration X increased, rate of generation X also increased.
Yes.
That's why I call it an activator.
Right. So in other words,
and this is, for example, this is inhibitor because G is a partial derivative of rate of generation Y by concentration Y. So if it has a negative value, if the concentration Y increase, the rate of generation Y decrease. That's why I call it an inhibitor.
And then you said if the diffusion coefficient of the inhibitor is bigger than that of the activator, then you get oscillatory patterns. Is that right?
Yes.
And otherwise it's stable, or you don't get. Then you have stable patterns.
Okay. If this determinant J is negative, this system is unstable to spatial fluctuation. And of course, this is square of square Q, so this is positive. And in order to make the system stable to temporal fluctuation, this should be positive. So this second term should be negative. So A11 dy plus A22 dx should be positive.
Oh, yes.
But
yes.
A11 plus A22 is negative.
I see, of course, yeah.
So that's why if one is positive, the other should be negative. So one is activator, the other is inhibitor. And in order to satisfy these two equations, dx should be smaller than dy.
Yeah, very good, yeah.
Now, the Planck model actually considers that the system is actually ideal. You're forming R and R, S and S. But we know that in practice, we know that once small peptides are formed, they form all kind of clusters again. And these clusters, as a matter of fact, control the ensuing process of the growth of this species. How, is it possible to introduce into this model also some kind of clustering after a while? We know in our case that if you have tetramers of DD, and LLL, they form a beta sheet, racemic beta sheet. And then the reaction continues not like isolated molecules, but they lower the energy of activation, and then you proceed to form very nice color. How this can be incorporated to your model?
Maybe just changing the K2 for this, and this is, because secondary nucleation process, new nuclei is generated on the surface of the minor crystal. So if something is attached to the minor crystal and inhibit generation of secondary nuclei, and it is enantiomerically selective, K2 for this process and this process should change.
So this mouse or mice business, that was the work of Suzuki, yeah? Or Suzuki 2003, was that when they had these traveling patterns on mice?
Mice, yeah. By Kondo and their co-workers did some work on traveling mice.
So this is most amazing. And it's really happening, yeah? and of course, octopus, for example, has also changes of colors and stuff, but this is very much controlled. Is that any way related in any sense?
It is not my job, so I don't know.
The zebra might be something. The zebra.
And even in the zebra and all these animals, how well is that actually understood? These are real chemical reactions in the skin or something like this.
Real animal is much more complex than the chemical reaction.
But maybe not
hard to predict it. And also,
I'm not sure, but maybe these are some kinds of chemical waves, but of activation, of the activation of genes. I think it's something like that, but I'm not sure. I think that you have some kind of stationary waves, but I don't know exactly what, if it's proteins or during the separation of cells. I don't know the details, but it's how genes are activated or not activated. Now we have communication between cells, but of course it's very, very complex.
So of course you.
I don't know.
Jim Murray's done a lot more on that.
Who?
Jim Murray.
Yeah, right. There's this book by Jim Murray, of course, on mathematical biology or something. Yeah.
Explains why very big creatures, very small creatures have uniform skin, medium-sized creatures that get punched.
Oh, is that right?
Creating mathematical model to generate chemical waves is not so hard. But what really happens to animal is very, very difficult.
In the living system, the nutrients or the chemical reactants or agency are transported in the blood, not by diffusion. Why there is pattern? Diffusion might not be so important. That's what I wonder. Why diffusion is the role?
Diffusion could not only real diffusion but also transport by convection or something like that. Every process is reversible.
Yeah, but for the pattern formation, the diffusion seems to be essential.
Maybe just simple communication from cell to cells. I don't know how it works, but we should have some specialists of things to move the answer to that.
So what goes through the blood? What did you say?
If the blood is if blood transforms everything, chemical.
Those pigments produced in the cell on the skin surface, not from the not transported from the blood.
It's not transported from the cerebroblast. So how is it transported then?
Just nutrition. There may be some enzymes, in the presence of enzymes, those kind of pigments are generated, and pigments doesn't live long, and maybe half-life not so long. So, in some part, generation of pigments are activated, but in some cases, some part, it is not activated. I don't know what scale we can say.
Any other questions, sir? Yeah, thanks very much then, it was great.